Hello, my name is Barua. I'm currently a PhD candidate in the Department of Chemical Engineering at Kansas State University. Welcome to my presentation on rapid screening and identification of microbial isolate collections using microbial recovery arrays. So microbes are ubiquitous single-celled microorganisms which exist in a microenvironment called microbiome by interacting with each other. And these interactions can be beneficial or detrimental. Understanding these interactions can be useful for production of engineered microbes, which can have beneficial impact on agriculture, human health, and bioenergy production. There have been applications of microbe interactions in production of high-value products and bioenergy. Also, scientists have been working on stimulating gut and lung microbiota for enhancing human health. There have been applications of different types of microbe interactions for soil bioremediation. And also there have been works going on for development of biocontrol agents and biofertilizers for enhancing plant health. And in my research, I'll be more focused on using such interactions for production of biocontrol agents and biofertilizers and enhance plant health. There have been many tools used for studying micro interactions. The most common one was traditional co-culture methods, where two different types of bacteria species are paired together on agar plates. There have been applications of 3D printing techniques where special compartments are used for isolating two bacteria species and understand the diffusive interactions among them. And also there have been uses of microfluidics which are capable of studying both dual species and multi-species interactions. And they can even go to single cell level, but uh, running this analysis is very difficult, and also they are mostly limited to on-chip analysis. In most of these methods, they are only limited to only two or few number of interactions. They are very qualitative in nature, and mostly they are time consuming and they are very low throughput, and they also cannot recreate complexities of natural microenvironments. Therefore, there needs to be developed a high throughput device that is capable of doing single cell studies, and also in which it is possible to control the microenvironment. That's why we have picked microwell recovery arrays, which are high throughput, high density wells fabricated on top of silicon wafers or glass substrates. So each of these arrays can contain tens of thousands of microwells, in which a focal species, which is fluorescently labeled, can be combined with other bacteria strains, and after that, the growth of the focal species can be tracked by using time-lapse fluorescent microscopy. So each of these will act as individual reaction sites where different types of interactions can be studied. In this paper, they have used two fluorescently labeled Pseudomonas aeruginosa strains for studying the interaction between them. So in my research, I'll be working on different types of micro interaction for the prevention of Agrobacterium tumefaciens. So Agrobacterium tumefaciens is a plant pathogen that causes crown gall disease in plants, and it has gained enormous economical importance because of the losses that US has to face each year for the tumors. And for that, we have picked a fluorescently labeled Agrobacterium tumefaciens species 15955 as the focal species, and we have combined it with the plant root isolates collected from Helianthus anus plants. And after combining these two types of bacteria in microwell arrays, it is our goal to screen the most potent inhibitors of Agrobacterium tumefaciens. In recent years, scientists have been targeting the three corners of crown gall disease triangle. The first approach was silencing plant genes which are required for propagation of Agrobacterium tumefaciens growth in plants. The second approach was controlling environment and preventing wounds by improving cultivation techniques in plants. And the third approach, which will be used by our research group, is to use pathogen isolates which are capable of inhibiting the growth of Agrobacterium tumefaciens in plants. So we have seen examples of such techniques used. In this paper, they have used agrocin producing Agrobacterium mutant strains to prevent the growth of Agrobacterium species 15955, and they did it on agar plates. In this research, they have used opine biosensor strains which are capable of reducing the size of tumors caused by Agrobacterium tumefaciens species 15955. And in this paper, they have showed that a human pathogen called Pseudomonas aeruginosa were capable of outcompeting Agrobacterium tumefaciens in um, biofilms because of quorum sensing regulated growth rate and motility advantages. Here, Pseudomonas aeruginosa is expressing red fluorescent protein, and Agrobacterium tumefaciens is expressing green fluorescent protein. Now I'll explain how we have used microwell recovery arrays for screening pathogenicity deficient Agrobacterium isolates capable of inhibiting the growth of Agrobacterium tumefaciens. We have microfabricated our microwells with a diameter of 10 micron in silicon wafers, and after that, the Agrobacterium isolates collected from Helianthus anus roots were combined with fluorescently labeled Agrobacterium tumefaciens SP15955 together in these wells to replicate Agrobacterium challenge model, which is favorable for the growth of Agrobacterium tumefaciens. We trapped them inside the wells using a photodegradable membrane, and we have tracked the growth of Agro 15955 for 24 hours based on the fluorescent signal. And after that, we found some wells which had diminished growth of Agrobacterium tumefaciens, and we targeted those wells as antagonistic outliers and exposed them to UV light for extraction of those cells by degrading the membrane above the target wells. After extracting those wells, we sequenced the isolates with 16S sequencing or whole genome sequencing to connect our microscopic observations with omics type characterizations. The photodegradable membrane that we have developed in our lab was a result of a Michael type cross-linking reaction between a polyethylene glycol crosslinker and polyethylene glycol thiol group in the presence of a pH 8 phosphate buffer. After the cross-linking reaction, there is a photodegradable O-nitrobenzyl diacrylate group between them, which can be exposed to UV light to cause photocleavage. And for causing that, we used a Polygon 400 pattern illumination tool, using which we could uh, expose our target well area in circle patterns or ring patterns to degrade the membrane above the target well and extract the cells out of those wells for validation and identification and characterization. Before we seeded our cells on top of microwell arrays, we at first had to identify which Agrobacterium isolates were extracted from Helianthus anus roots. And for that, we actually took out 481 root samples and then washed them in ATGN liquid media and then plated them onto yeast lactose media and ATGN to streak isolate a total of 309 Agrobacterium isolates. And after that, we sequenced these 309 Agrobacterium isolates, and we found out that all of these isolates were unique in terms of genetic characteristics. And then we use these Agrobacterium isolates to combine with Agro 15955 for our co-culture experiments and monoculture experiments in microwell arrays. We cultured our Agrobacterium isolates and Agro 15955 in AT minimal media, and then we seeded Agro 15955 in monoculture at an optical density of 0.1 on the microwell arrays, and we found out that the cells grew uniformly all over the wells. But there were some outlier wells where cells didn't grow as well as the others. It can be due to lower nutrient availability in the wells, and also it can be due to having less cells to begin with in the culture. Then we co-cultured Agro 15955 and these Agrobacterium isolates at a ratio of 1 to 1, 10 to 1, and 100 to 1. The goal of using three different ratios was to reach a ratio where the growth conditions are more favorable to the growth of Agro 15955. And we found out that there were very high range of variations in growth characteristics in the ratios of 1 to 1 and 10 to 1. But with 100 to 1, we got almost similar growth trends as Agro 1595 monoculture. And we also found a lower number of outliers. A point should be noted is that in case of monoculture, these outliers can be due to lower nutrient availability. But in co-cultures, these outliers could be due to the presence of different types of isolates which are inhibiting the growth of agro 15955. The only way to know this is to extract these cells out of these wells and sequence them to find different types of genetic variations which are responsible for the diminished growth of agro 15955. To support our claim that 100 to 1 ratio is the best ratio for generating agrobacterium challenge model, we uh, analyzed the growth rates of agro 15955 in all of these wells, and we found out that the growth rates in the ratio of 1 to 1 and 10 to 1 was very low because of different types of growth variations, but at 100 to 1 and co-culture, we found similar growth rates. We also found that the number of outliers present in 1 to 1 and 10 to 1 ratio were significantly higher than the outliers present in 100 to 1 ratio and agro 15955 monoculture. That's why we could say that 100 to 1 is the best ratio favorable for agro 15955 growth. And we have used this co-cultured wells for extracting the most potent inhibitors of agro 15955. So after these antagonistic outlier wells are targeted, we expose them to UV light. So we exposed UV light in a ring pattern. So the wells have a diameter of 10 micron and the diameter of the UV ring pattern was 20 micron. And after that, we extracted the cells out of the wells by washing them with AT minimal media and streak isolated them in ATGN agar plates to get 9 individual isolates from 3 microwells extracted. After extracting these 9 individual isolates, we had to validate the claim that these individual isolates were capable of inhibiting the growth of agro 15955. And we validated this claim using a 96 well plate reader assay. For that, we cultured each individual isolate in ATGN liquid media and collected the cell-free culture media from each of these individual cultures. We also combined the cell-free culture media from all individual cultures in equal volume to generate 9-member consortia effect. After that, all of this cell-free culture media was combined with a fresh culture of agro 15955, and then we tracked their growth in 96 well plate reader. We also tracked the growth of agro 15955 in monoculture in fresh ATGN media. And then we used an R package called growth curver to determine the growth rate and carrying capacity of agro 15955 in each of these combinations. So growth rate is the amount of cells growing in each hour, and the carrying capacity is the highest optical density of cells in this culture before reaching stationary phase. And after that, we determine the growth rates and carrying capacities of each of these combination at n equal to 6 replications. And we, while comparing their growth rate and carrying capacity with the growth rate and carrying capacity of agro 15955 in monoculture, we found out that the growth rate and carrying capacity of agro in all individual cell-free culture media and in the 9-member consortia culture media were significantly lower than the growth rate and carrying capacity of agro 15955 in ATGN media. From this, we could validate the claim that each of these individual isolates, individually or as a consortia, could inhibit the growth of agro 15955. After this, we have to further validate our claim by doing inhibition assay of this antagonistic agrobacterium isolates in ATGN agar plates. We also need to identify the agrobacterium isolates responsible for agro 15955 antagonism with the help of 16S sequencing and whole genome sequencing. Currently, we're in the process of studying whole genome sequence data for determining gene clusters which are responsible for inhibiting the growth of agro 15955. So these are all I had for today. Thanks a lot for listening to my presentation. If you have any question, please let me know. Thank you.